All right, time to go teach. Hello and welcome to this next episode of Tutors, where we are continuing on biochemistry, and today we are going to look at glucose polymerization. Basically, what that means is we are going to focus on only one type of monomer, one monosaccharide, and that's going to be glucose, and the different polysaccharides that monomer can make. So here we go, targets of the day. First is identify the different types of glucose polymers. So even though we're only using one monomer, it can form different types of polymers. Next, we're going to look at the different functions of each of those glucose polymers, because since they all have a different shape, since all of these glucose polymers have a different shape, that means they are all going to have a different function. Structure and function always go hand in biology. And lastly, we're going to compare and contrast those different types of glucose polymers, making sure that we understand what ways to identify them, what they're used for, and how they are structured. Let's go and look at it. First off, we need to recognize that there are actually two different isomers of glucose that we're going to be looking at. The first type of glucose right here, this is alpha glucose, and the second one over there is beta glucose. The only difference between each of these glucoses is this group right here and this group right there. It is a hydroxyl group, and the way that the hydroxyl and the hydrogen are arranged around this specific carbon. Now, to actually look at all of this, one of the things that we're going to have to be able to do is count our carbons. And to count carbons, we need to be able to identify which carbon to start with and which direction to then go. So here's how it's done. We're going to look at our alpha glucose first. And the part of this ring that we're going to look at originally is going to be our oxygens that happen to be connected to this singular carbon. The reason we're going to look at those two oxygens are because each other carbon is only attached to one other oxygen. So here, this special carbon is the only one that is attached to two oxygens. And because of that, we are actually going to use it, and we're going to count that carbon as being the primary carbon. We're also going to refer to that as the first carbon. So we will give it the designation of a 1. And then we are just going to work our way around the ring. We can't go up because that would take us to an oxygen, and that doesn't help us counting carbons. So we are just going to go in a sequential order right around. So carbon 2, carbon 3, carbon 4, carbon 5, and lastly, this is the 6th carbon. So now we know and we can identify each of our carbons based upon which number they have. If I'm talking about the carbon 1, that's what I'm talking about. If I talk about carbon 6, I will be talking about that one always. The important carbons that we're going to talk about in this lesson are going to be the first carbon, the 4th carbon, and the 6th carbon. Those are the primary carbons that we are going to be discussing today. But just so you know, we do count the carbons like this for every single one of our monosaccharides. It's very traditional. This is a very standard practice on how we determine which carbon is which. So that way, when I identify a carbon, you understand exactly which carbon I'm talking about. So, alpha glucose. As you know, when these monomers come together, they are going to undergo dehydration synthesis. So we're going to bring in another alpha glucose. And right here, you can see we have the first carbon, and it is right across from this glucose 4th carbon. And it's their hydroxyl groups that are going to interact and form our dimer. So now we have two glucose molecules bonded together across the 1st and the 4th carbon. And we can do this multiple times. We can continuously bring in a brand new alpha glucose, and again, water's released, and it is the 1st and the 4th carbon that are being involved in these different reactions. It doesn't matter how many we do. Every single time we're looking at the first and the fourth carbon of these different glucose molecules interacting to form this bond. And this happens to be the polysaccharide starch, where we have an alpha 1,4 linkage. We call it an alpha 1,4 linkage because it is for alpha glucose and it is across the first and the fourth carbons. This is also referred to as a glycosidic linkage. So, starch. What is it? How does it work out? Well, first off, starch is made by plants and it is used for energy storage. It is only made using glucose. It is 100% glucose, and they use that 1,4 glycosidic linkage. It is an alpha 1,4 again because it uses alpha glucose. And that is the polysaccharide starch. We're going to do a little modification to this to see how do animals use glucose. So we're going to not look at starch for a second. We're going to actually look at another polysaccharide called glycogen. Now, glycogen, it's used for energy storage for animals. And what occurs here is that one big difference. And that is that now we've just taken an alpha glucose and we've just turned it. This is the first carbon, second, third, fourth, fifth, and sixth. So 1, 2, 3, 4, 5, 6. Those are the same as what they've always been. And now it is the first carbon facing the sixth carbon of this other alpha glucose. And their hydroxyl groups are going to interact. And we have dehydration synthesis. And now we have an alpha 1,6 linkage. And this molecule of glucose right here can now continuously do an alpha 1,4, or it also could do another 1,6 linkage, depending on how these glucose molecules are put together. It all depends on the reaction that's taking place. So glycogen is a little bit different from starch, because starch, well, it's mostly this straight chain. Starch can actually branch off in that alpha 1,6 linkage, but for the most part, it's in these 1,4 linkages making a straight chain. Glycogen, on the other hand, it branches at that alpha 1,6 a lot of times. So instead of being a straight chain, it's going to end up being this network, this web that comes out and is a very branched system. So starch, when we think about it, we generally think of starch in high school biology classes as being a straight chain, and glycogen as being a highly branched chain. Even though starch does make some of those 1,6 linkages, it doesn't make nearly the amount that glycogen makes. So when we look at them, glycogen, hey, it's made, again, by animals. We put our glucose together as glycogen. It's a polysaccharide also of alpha glucose, so it uses the exact same molecule that starch uses, except it is highly branched, whereas starch is basically a straight chain. Because the glucose is linked in both a 1,4 and the 1,6 glycosidic linkage, between 8 to 12 units of glucose that are bonded together in a 1,4 linkage, an alpha 1,6 is also going to be formed. So they're going to branch about every 8 to 12 units. Starch, very much straight chains, and glycogen, highly branched. Both using alpha glucose, one having a lot of 1,6 linkage, the other one mostly just the 1,4. But there's that other type of glucose. It's beta glucose. So what gives? What about beta glucose? What makes it different and how is it used? Well, again, the difference here is how that hydroxyl group is coming off the first carbon. And that's going to mean that when it bonds using dehydration synthesis with another beta glucose, they're not going to bond straight across. In fact, what's going to happen is we need to flip our other beta glucose. And there we go. Now, this is still a 1,4 linkage. This is our 1 carbon, and this is our 1, 2, 3, 4 carbon. So it still is a 1,4. But instead of being an alpha 1,4, this is a beta 1,4 linkage. And then we can bring in another beta glucose, and that beta glucose will come in and bond, and it's still a 1,4 linkage. Here is our 1 carbon, and then here is our 4 carbon. And so we are going to continuously have this chain, but it's 1,4. It's beta 1,4 linkages, and we can see that the 6 carbon alternates facing up, facing down, facing up, facing down. So the orientation of our glucose molecule is going to constantly flip. Where is it used? Cellulose, in plants. Beta glucose is used to make plant cell walls. It makes an extremely rigid structure. These beta 1,4 glycosidic linkages are interesting because they make it much harder to be broken down. The polysaccharide cellulose is extremely hard to hydrolyze or to perform hydrolysis on. That's why it provides such great structural support. The reasoning behind it really is because our chains of glucose end up being able to hydrogen bond with each other. So you're going to have layers and layers and layers of these chains that are hydrogen bonding together, and that makes it extremely strong of a structure, which is good for all of our plants that need to have a strong and rigid cell wall. And that takes us to the summary. That's it, guys. There are only 3 very important polymers that are made entirely of glucose. Starch and glycogen, which are both used for energy storage. The big difference between them is that glycogen is highly branched because it uses the 1,4 linkage and the 1,6 linkage, whereas starch is mostly just that 1,4 linkage, so it mostly is just these straight chains. Cellulose, on the other hand, it doesn't use the alpha glucose, it uses a beta glucose. Beta glucose forms those 1,4 glycosidic linkages. It does it in a flip-flop orientation, and that means that those different chains of glucose are going to be able to hydrogen bond, and that's going to give them extremely strong structural stability. So those are the 3 different polymers that you can make from glucose. That's it for this time. Till next time, be awesome, stay awesome.